Hello everyone, I am Dr. Pokhraj Guho, Assistant Professor, Department of Zoology, Garbeta College. Welcome to my course on the basics of the immune system. Today I will speak on the first core topic, that is core 1 of my course, named cell and organs of the immune system.
In the first line, you can see that the following figure depicts different organs that are related to the immune system. It includes the adenoids, the tonsils, the thymus, the bronchus associated lymphoid tissue, BALT part, bone marrow, intestine, Peyer's patches, appendix, inguinal lymph nodes, spleen, axillary lymph nodes. The immune system, you can see that it is a host defense system. What is a host defense system? Means it comprises of a number of organs, a number of cells that works in coordination with one another and protects the system against any pathogens that enter our body, maybe from viruses, bacteria, parasitic worms, etc. And secondly, it distinguishes our own self antigens and self molecules with that of foreign molecules or those molecules that try to enter our body, and thus by differentiating self from the non-self, it protects our body from any type of foreign invasions. It is classified into subsystems. One type of subsystem is the innate immune system and the adaptive immune system. The innate immune system that we gain during our birth, right from our birth, it is more or less non-specific. And on the other hand, we have the adaptive immune system. The adaptive immune system comprises of that part of the immune system that we gain through our exposure to different antigens during our lifetime, and this is a sort of specific type of specific wing of the immune system. On the other hand, another subsystem exists which says about the humoral immunity and the cell-mediated immunity. What is humoral immunity? Humoral immunity depicts the part of the immune system that is controlled by the B cell, and the antibodies that are secreted by the B cell. And the cell-mediated immunity comprises of the T cells, and the way the T cells, cytotoxic T cells are there, CD8 T cells. These T cells protect our body by destroying the infected or the damaged cells. In human, the blood-brain barrier, blood cerebrospinal fluid barrier, and fluid-brain barriers are there, which can separate the peripheral immune system from the neuroimmuno system, which protects the brain. The organs of the immune system can be of two types. They are the primary lymphoid organ, which provide appropriate microenvironment for the development and maturation of the lymphocyte, which includes the thymus, bone marrow, and fetal liver. And the secondary lymphoid organ, which are responsible for trapping antigens, and these antigens are then showed to the mature lymphocyte, which can interact effectively with that antigen. It means the antigen, the lymphocytes get activated in the secondary lymphoid organ. These are the spleen and the lymph node, the MALT, the mucous-associated lymphoid tissue, gut-associated lymphoid tissue, and lastly the bronchial associated lymphoid tissue. You can find another type of lymphoid tissue, that is a tertiary lymphoid tissue, which is the most prominent example is the COLD, or the cutaneous associated lymphoid tissue. These store fewer lymphoid cells than that of the secondary lymphoid organs, but can import lymphoid cells during an inflammatory response and can provide immediate protection. Now, a large number of cells are involved in the immune system. It includes the neutrophils, polymorphonuclear neutrophils, you all know, the lymphocytes, B and the T lymphocytes, natural killer cells, eosinophils, and antigen presenting cells. The immune system consists of two main groups of cells: the lymphocytes, which are mainly the effector cells and provide protection by directly interacting with the antigen, and the antigen presenting cell, which are responsible for presenting the antigen and making them recognizable by the lymphocytes. The lymphocytes are mainly responsible for initiating adaptive immune response in the human body. All lymphocytes are produced in the bone marrow system by a process called as hematopoiesis. Two main lymphocytes are there, types of lymphocytes are there. They are the T lymphocytes and the B lymphocytes. Now, I have mentioned about the hematopoiesis, and it occurs in a special kind of cell. They are known as the hematopoietic stem cells. Stem cells are cells that can differentiate into other cell types. As you all know, they are self-renewing, they maintain their population's level by cell division. In this diagram, you can see that the stem cell can differentiate into proerythroblast, myeloblast, lymphoblast, monoblast, and megacardioblast. Megacardioblast can give megacardioblast can give rise to the platelets. The monoblast can give rise to the monocyte. The lymphoblast to the lymphocyte, that is the T and the B lymphocyte. And myeloblast, it can give rise to all the sorts of granulocyte, basophil, eosinophil, and neutrophil. And lastly, the proerythroblast, it gives rise to the red blood cell. So there are a few differences between B lymphocyte and T lymphocyte. B lymphocyte originate and they mature in the bone marrow, whereas the T lymphocyte originates in the bone marrow but matures in the thymus. Each B cell is genetically programmed to have a specific B cell receptor, which is nothing but an antibody molecule, membrane-bound antibody molecule. And in the other hand, T cell has specific receptor in its membrane termed as the T cell receptor. B cells can produce antibodies, that is produced by the plasma cell. Plasma cell is a differentiated type of B cell, and it can also give rise to the memory cells. Whereas T cell can only recognize antigen if it is bound to major histocompatibility complex.
Let's have a tune discussion.
Where in birds do B cells develop?
The bursa of Fabricius, or cloacal bursa, is the site of B lymphocyte B cell maturation and is unique to birds. It is a dorsal outpouching of the proctoderm, or most distal portion of the cloaca. The bursa develops before hatching and reaches its maximum size by several weeks of age. It is found to be important in the maturation of B lymphocytes, B for bursa. The equivalent in humans is probably the tonsils or lymphoid tissue in the intestine.
How old is the concept of immunity?
The first vaccine was developed in the late 18th century, but people recognized the importance of immunity long before that. During the plague of Athens in 430 BC, the Greeks realized that people who had previously survived smallpox didn't contract the disease a second time. In fact, these survivors were often called upon to attend to those afflicted with smallpox, according to a 1998 article in the International Journal of Infectious Diseases. In the 10th century, Chinese healers began blowing dried smallpox scabs into the noses of healthy patients, who then contracted a mild form of the disease, and the patients who recovered became immune to smallpox. This practice, which was called variolation or inoculation, spread to Europe and New England in the 1700s.
Thank you for your patient hearing. Visit me at pokhrajclasses2022.genomeo.com or call me at 977 599 5422.